Shalom and welcome to the Hebrew of Israel channel. This episode will cover the three sons of Noah from a genetic perspective. According to the scriptures, Noah had three sons named Shem, Ham, and Japheth, and only 8 lives were saved on the ark. And it's up to them to repopulate the world.
These are the generations of Noah. Noah was a just man and perfect in his generations, and Noah walked with God. And Noah begat 3 sons, Shem, Ham, and Japheth. And the sons of Noah that went forth of the ark were Shem, and Ham, and Japheth. And Ham is the father of Canaan. These are the 3 sons of Noah, and of them was the whole earth overspread.
There is a strong parallel between Adam and Eve and Shem, Ham, and Japheth, as Adam and Eve were the father and mother of the pre-flood world and had dominion over it. Shem, Ham, and Japheth became the new fathers, and their wives the new mothers of this post-flood world, and are given dominion over it.
And God said, Let us make man in our image, after our likeness, and let them have dominion over the fish of the sea, and over the fowl of the air, and over the cattle, and over all the earth, and over every creeping thing that creepeth upon the earth. So God created man in his own image. In the image of God created he him. Male and female created he them. And God blessed them, and God said unto them, Be fruitful, and multiply, and replenish the earth, and subdue it, and have dominion over the fish of the sea, and over the fowl of the air, and over every living thing that moveth upon the earth. And God said, Behold, I have given you every herb bearing seed which is upon the face of all the earth, and every tree in the which is the fruit of a tree yielding seed. To you it shall be for meat. And to every beast of the earth, and to every fowl of the air, and to everything that creepeth upon the earth, wherein there is life, I have given every green herb for meat. And it was so. And God saw everything that he had made, and behold, it was very good. And the evening and the morning were the 6th day. Right. And God spake unto Noah, saying, Go forth of the ark, thou, and thy wife, and thy sons wives with thee. Bring forth with thee every living thing that is with thee of all flesh, both of fowl and of cattle, and of every creeping thing that creepeth upon the earth, that they may breed abundantly in the earth, and be fruitful, and multiply upon the earth. And Noah went forth, and his sons, and his wife, and his sons' wives with him. Every beast, every creeping thing, and every fowl, and whatsoever creepeth upon the earth after their kinds, went forth out of the ark. Chapter 9. And God blessed Noah and his sons, and said unto them, Be fruitful, and multiply, and replenish the earth. And the fear of you and the dread of you shall be upon every beast of the earth, and upon every fowl of the air, upon all that moveth upon the earth, and upon all the fishes of the sea. Into your hand are they delivered. Every moving thing that liveth shall be meat for you. Even as the green herb have I given you all things. But flesh with the life thereof, which is the blood thereof, shall ye not eat. And surely your blood of your lives will I require. At the hand of every beast will I require it, and at the hand of man. At the hand of every man's brother will I require the life of man. Whoso sheddeth man's blood, by man shall his blood be shed. For in the image of God made he man. And you, be ye fruitful, and multiply. Bring forth abundantly in the earth, and multiply therein.
I believe all of mankind today can trace their lineage back to Noah's 3 sons, Shem, Ham, and Japheth. Remember, the flood was global and killed off all humans except for the 8 on the ark. This is why the Most High told Shem, Ham, and Japheth to repopulate the earth, which is the same words the Most High told Adam and Eve. Again, Noah and his sons are like a new Adam and Eve.
And God blessed them, and God said unto them, Be fruitful, and multiply, and replenish the earth, and subdue it, and have dominion over the fish of the sea, and over the fowl of the air, and over every living thing that moveth upon the earth. And you, Be ye fruitful, and multiply. Bring forth abundantly in the earth, and multiply therein. These are the 3 sons of Noah, and of them was the whole earth overspread.
According to Genesis chapter 10, Shem, Ham, and Japheth had many children.
Chapter 10. Now these are the generations of the sons of Noah, Shem, Ham, and Japheth. And unto them were sons born after the flood. The sons of Japheth, Gomer, and Magog, and Madai, and Javan, and Tubal, and Meshech, and Tyrus. And the sons of Gomer, Ashkenaz, and Riphath, and Togarmah. And the sons of Javan, Elisha, and Tarshish, Kittim, and Dodanim. By these were the isles of the Gentiles divided in their lands, every one after his tongue, after their families, in their nations. And the sons of Ham, Cush, and Mizraim, and Phut, and Canaan. And the sons of Cush, Seba, and Havilah, and Sabta, and Raamah, and Sabteka. And the sons of Raamah, Sheba, and Dedan. And Cush begat Nimrod. He began to be a mighty one in the earth. He was a mighty hunter before the Lord. Wherefore it is said, even as Nimrod the mighty hunter before the Lord. And the beginning of his kingdom was Babel, and Erech, and Akkad, and Calneh, in the land of Shinar. Out of that land went forth Asher, and builded Nineveh, and the city Rehoboth, and Calah, and Resen between Nineveh and Calah. The same is a great city. And Mizraim begat Ludim, and Anamim, and Lehabim, and Naphtuhim, and Pathrusim, and Cashluhim, out of whom came Philistim, and Caphtorim. And Canaan begat Sidon, his firstborn, and Heth, and the Jebusite, and the Amorite, and the Girgasite, and the Hivite, and the Archite, and the Sinite, and the Arvadite, and the Zemarite, and the Hamathite. And afterward were the families of the Canaanites spread abroad. And the border of the Canaanites was from Sidon as thou comest to Gerar unto Gaza, as thou goest unto Sodom and Gomorrah, and Admah, and Zeboim, even unto Lasha. These are the sons of Ham, after their families, after their tongues, in their countries, and in their nations. Unto Shem also, the father of all the children of Eber, the brother of Japheth the elder, even to him were children born. The children of Shem, Elam, and Asher, and Arphaxad, and Lud, and Aram. And the children of Aram, Uz, and Hul, and Gether, and Mash. And Arphaxad begat Salah, and Salah begat Eber. And unto Eber were born 2 sons. The name of one was Peleg, for in his days was the earth divided. And his brother's name was Joktan. And Joktan begat Almodad, and Sheleph, and Hazar-Maveth, and Jerah, and Hadoram, and Uzal, and Diklah, and Obal, and Abimael, and Sheba, and Ophir, and Havilah, and Jobab. All these were the sons of Joktan. And their dwelling was from Mesha, as thou goest unto Sephar, a mount of the east. These are the sons of Shem, after their families, after their tongues, in their lands, after their nations. These are the families of the sons of Noah, after their generations, in their nations, and by these were the nations divided in the earth after the flood.
I believe all of current humanity can trace their lineage back to the people mentioned in Genesis chapter 10. And I believe DNA can prove this. The scriptures gives us a few scientific tools we can use to determine this. First of all, Genesis chapter 10 mentions men for the descendants of Shem, Ham, and Japheth. Now obviously, Shem, Ham, and Japheth had daughters. This is even mentioned in the scriptures. But the focus seems to be on the male lineage. Scientifically, we call this a paternal lineage. We can use Y-DNA haplogroups to figure out the lineage of Shem, Ham, and Japheth. But before we go too deep, what is a Y-DNA chromosome? Copyright disclaimer. Under Title 17 U.S.C. Section 107, allowance is made for fair use for purposes such as criticism, comment, news reporting, teaching, scholarship, and research. Fair use is permitted by copyright statute that might otherwise be infringing. Non-profit educational or personal use tips the balance in favor of fair use.
As many Americans know, the third president of the United States and author of the Declaration of Independence, Thomas Jefferson, was an important figure in the emergence of early American identity and is considered one of the major founding fathers of the country. And although many today argue over his own personal moral character and ethics, it was a major point of contention even hundreds of years ago, and many spread rumors of Jefferson's ongoing affair with his slave Sally Hemings, and whether he was the father of her 6 children, an act which would have been highly frowned upon even in the 18th century. Centuries later, in 1998, it has seemingly been proven through DNA tests that Jefferson was indeed the biological father of Hemings' children, despite the fact that there were no viable biological samples of the deceased president in existence. So how did they do it? When it comes to human genetics, perhaps the best way to determine hypodescent or common ancestry in individuals and population clusters is through a specific genetic marker known as a haplogroup. Haplogroups are a common way for geneticists, archaeologists, and historians to make conclusions over the origins, exchange, and evolution of people groups all over the world, and you may have heard me mention them in videos from time to time for the past year, but unless you knew beforehand, information on haplogroups may just cause confusion and misinformation for many viewers. Therefore, I've created this video to better give an understanding of how haplogroups relate to genetics, race, ethnicity, community, and other aspects of humanity that paint such a beautiful picture of the history and diversity of our planet. For as long as can be traced back, all females carry a maternal haplogroup inherited from their mother, while males carry both a maternal and paternal haplogroup, seeing how men have an X and Y chromosome. The paternal haplogroup inherited by the father is known as the Y haplogroup from the Y chromosome, while the maternal haplogroup is inherited from mitochondrial DNA and is thus referred to as the mtDNA. Because of historical patterns of migration and the nature of male-female relationships for most of human history, generally speaking, paternal haplogroups are better studied, understood, and mapped out, although this doesn't make maternal lineage irrelevant or any less interesting. For this reason, haplogroups will only survive to the next generation if they're passed on to their offspring of the same sex through the X or Y chromosomes. Now, you may not necessarily ever have an offspring that is the same gender as yourself, in which case your specific haplogroup would die with either you or your child, but at least one of your parents sure did, as with one of their parents and grandparents and great-grandparents and so on, back to the genesis of the human species. Barring genetic mutations, if you're a male, it's an absolute certainty that you will share the same paternal haplogroup with your father, all of your biological brothers born of the same father, all of your paternal uncles, any paternal male cousins born to your uncle, and of course your paternal grandfather, his father, and so on, and all biological male children of yours will share your paternal haplogroup. Similarly, barring genetic mutation, regardless of gender, you will share the same maternal haplogroup with your mother, all of your biological siblings born to the same mother, all of your maternal aunts and uncles, any maternal cousins born to your aunt, as well as your maternal grandmother, her mother, and so on. And if you're a female, then all biological children of yours will share your maternal haplogroup, although only your female children will pass it on to their offspring. Now, you may be wondering, how did the first haplogroups form? What is the criteria for categorizing various haplogroups? How and why did the haplogroups we see today become scattered across the planet? All good questions, and as usual, the simplest explanation is often the correct one, as indeed all living humans today share ancestry with a common ancestor in both the male and female line, dubbed the genetic Adam and Eve of the human species, and the descendants of these two evolved into the 7.6 billion people of Earth we see today, and the genetic adaptations and natural selection form the modern races of humanity. New haplogroups are created when slight mutations occur when passing DNA to the next generation, and these mutations can generally be traced back to the original source, and hence the standard naming convention for haplogroups is a letter of the English alphabet, followed by the ordering of whichever specific group or subclade its mutation belongs to. In conclusion, haplogroups, along with language, ethnicity, and religion, are one of the best ways of accurately defining population groupings of the human species, and I hope this better explained haplogroups, DNA, and human genetics, and showed you just how interconnected the world's population is, even if every group has its own unique traits, history, and culture. Please let me know.
What's up, everyone? I'm Helena Alamu, and welcome to another episode of the DNA Download, where we explain the science of genetics so you can live in the know. When people talk about their DNA results, I keep hearing the word haplogroups. Joining me today is Dr. Samantha Esselman, a scientist from the 23andMe Ancestry team who can unpack this concept. So, Samantha, what are haplogroups? Truly, it sounds like a word my dad would make up in Scrabble.
Well, it's definitely a real word. A haplogroup is a grouping or an assignment of similar sets of patterns in people's DNA. So it's kind of like if we were to group people based on something like their zip codes. And then we can use these patterns of DNA to trace the ancient origins of your ancestry.
Okay, so then how do you make these groupings from DNA?
So people have two special pieces of DNA which we use to define two different kinds of haplogroups. The first kind is paternal haplogroups traced through Y chromosomes, which get passed exclusively from fathers to sons. And the second kind is maternal haplogroups traced through mitochondrial DNA, which gets passed from mothers to both sons and daughters.
Okay, then what makes these two pieces of DNA special?
So that's a great question. It's because the inheritance pattern is really simple, especially compared to the rest of your genome. And actually the beginning of the word haplogroup, haplo, comes from the Greek word aplos, which means simple.
Oh, well, that's a good memory jogger.
Yeah. And so most of our DNA comes from a sort of random shuffling of our parents' DNA, which can make it difficult to figure out which side of the family each piece comes from.
Oh.
But then Y chromosomes and mitochondrial DNA don't get shuffled.
But then if they aren't shuffled, why aren't everyone's haplogroups exactly the same?
That's a great question. So over the more than 200,000 or so years of human evolution, the occasional DNA changes occurred and got passed on, creating new haplogroups.
Okay.
I have an analogy.
Okay.
So imagine a guy thousands of years ago, and this guy has a special recipe for his, award-winning paleo soup.
Oh, okay.
And then he, along with his brothers, inherited this recipe from his dad, who got it from his dad, and all of the men in the family faithfully stick to this recipe, no changes.
Oh, well, I could go for some paleo soup right about now, but please, go on.
Yeah, this analogy always makes me hungry.
Okay, good.
And so then many generations later, a great-great-great-grandson adds some cilantro. And then this great-great-grandson passes on a slightly modified version of this recipe to his sons. And all the men on his branch of the family tree start following this new cilantro recipe.
Oh.
And so now if you think about it, there are essentially two soup haplogroups. And this is pretty much how haplogroup inheritance works.
That makes a lot of sense. And since I'm a woman and I don't have a Y chromosome, how can I trace my paternal haplogroup?
Yeah, so what you can do is you can learn from relatives who share your paternal line. And so this would be something like a dad or a brother.
Okay, cool. So then what about the mother's side, the maternal haplogroups?
Right, you can trace this other kind of haplogroup through the DNA found in our cells' mitochondria, which turn the food we eat into usable energy for our cells.
Ah, the powerhouse of the cell.
You know your biology memes. Yeah, so this mitochondrial DNA gets passed from moms to both sons and daughters.
Well, thanks, Mom. So how can we use haplogroups to trace our ancestry?
So closely related haplogroups tend to share geographic roots, and so scientists can use the kind of modern distributions of haplogroups around the world to trace some of these major migrations. And then these can help you understand where you fit into this ancient family tree of all humans.
Wow, I feel like the world just got a whole lot smaller. Thanks for being here, Samantha, and see you next time on the DNA Download.
When you get your DNA tested for genealogy, you will often see the term haplogroup come up. What is that? Join me on today's Ancestral Findings podcast, and I'll talk on what is a haplogroup and how does it pertain to your family tree. If you get your DNA tested, chances are you will come across the term haplogroup. What is a haplogroup, and how does it pertain to your family history? At its essence, a haplogroup is an ancestral clan. Some clans are the Vikings, Native Americans, all tribes, Celts, Aboriginal Australians, and other such groups. Your haplogroup tells you where your ancestors came from deep back in time. There are also male and female haplogroups, so you can see where your male and female sides of the family originated back in prehistoric times. As with Y DNA, which traces the male line from father to son, and mtDNA, which traces the female line from mother to daughter, haplogroups also follow straight male and female descendancy lines. Your haplogroup's point of origin is usually shown on maps at the various DNA companies that do testing. The haplogroups that exist today originated with people whose descendants were very successful at reproducing in large families. Haplogroups today are divided into four main ones: European, African, Native American, and Asian. Within these haplogroups are many subhaplogroups that further define where a person's earliest known non-African ancestor on the male and female sides of their family originated, and when. Most haplogroup subgroups are plentiful. The only group with less than 3 subgroups is the Native American male group, which only has 2 subgroups. Basically, haplogroups chart the clans from which the modern human race originated. Haplogroups are named alphabetically in order of discovery. The subgroups of haplogroups are named with letter and number combinations, indicating where and when in time they were discovered, which main haplogroup they descended from, and sometimes even the very specific small geographical area where their earliest members lived. The more letters and numbers in your haplogroup's subgroup, the more likely you are to be able to pinpoint an exact region, and maybe even a county or town. Some haplogroup subgroups can even be traced back to named individuals if the mutation that created them originated in the more recent past. For example, the infamous Celt, Niall, of the 9 Hostages, is known to be the originator of a subgroup of a haplogroup within the past 1,500 years. And some people can use their subgroup to prove their descent from him. Right now, haplogroups are the only way to trace your family tree back to the time before surnames were invented. You can take your last known ancestor in a particular direct male or direct female line and connect them to a larger clan in the more distant past, knowing that their ancestors, the ones you can't identify and trace, wound back through time to the time and place where the known ancestor's haplogroup and subgroup originated. For example, there is a known haplogroup of people who originated in France about 12,000 years ago, and who are widely believed to be the people who made the famous cave paintings in the French town of Chauvet. You can look at a maternal or paternal haplogroup and subgroup and see if it matches with the haplogroup and subgroup of the cave painters. If it does, you know you are descended from this illustrious and famous group of people. If you're interested at all in where your family originated deep in time, before the age of records, then haplogroups should be of great fascination for you. You can use them to get deep into history with your family tree. This is something that was not possible before the age of DNA testing.
Lineage important in the modern day? Some say no, but biology says yes. In almost every single culture around the world, much emphasis was placed on lineage, which is referring to an individual's line of ancestors, and still is important to a certain extent today. Some people around the world are quite proud of their lineage, and others not so much, while others say it shouldn't matter at all. Various cultures have different naming customs passed down from generation to generation in order to preserve their lineage, and this may vary by country. For instance, in the Anglo world, traditionally the father would pass on his surname to his children, but in the Spanish-speaking world, both the mother and father pass on their surnames to their children. Some cultures may traditionally only have one given name, while others may have several, but the surname, or family name, is what is generally passed on to cement the lineage of a certain family. The Kennedy, Clinton, Bush, Roosevelt, and Adams families are all examples of political families in the U.S. that have arguably been successful due to the power of their lineages. There is certainly power in a name. Nature thinks so too. That's why each and every human on the planet has a genetic surname. They're called haplogroups. Haplogroups are genetic markers that are extremely important in the study of history and anthropology because they can show an individual's or collective's simplified genetic past that gives clues to how different populations interacted. I've explained this in a past video, but essentially a woman will pass down her mitochondrial or maternal haplogroup onto all of her children, while a man will only pass down his Y DNA or paternal haplogroup only to his sons, because only males have a Y chromosome. Your maternal haplogroup is passed down from your biological mother to you, and it was passed down to her by her mother, your maternal grandmother, and your grandma's mom, and so on and so on. Since only males have a Y chromosome, if you were male, your father passed his paternal haplogroup to you, passed down by his father and his father before him. Are you starting to see how this mirrors many of the naming customs from around the world? Geneticists who are far more knowledgeable than myself on the subject have been able to map out haplogroup lineages around the world going back tens of thousands of years, which gives massive insight into past human migrations, periods of intermixing, and conquest. For tens of thousands of years, these haplogroups have been spreading across the globe, splintering and diversifying due to genetic mutation between generations, and this paints a very interesting picture. This is a pretty awesome map showing paternal haplogroup distribution around the world, but I think
So as we can see, Y DNA haplogroups are a perfect tool we can use to determine the male lineage. We can use this to find the descendants of Shem, Ham, and Japheth, because haplogroups are passed down from father to son. This is exactly what we need to know. According to livingdna.com, what are haplogroups? It says, the paternal haplogroup relates to your Y chromosome, and since that is the sex-determining chromosome for men, it is passed down from father to son. Now the next method to use in determining the descendants of Shem, Ham, and Japheth is language. After all, it was language that was used to separate and divide the descendants of Shem, Ham, and Japheth.
By these were the isles of the Gentiles divided in their lands, every one after his tongue, after their families, in their nations. These are the sons of Ham, after their families, after their tongues, in their countries, and in their nations. These are the sons of Shem, after their families, after their tongues, in their lands, after their nations. These are the families of the sons of Noah, after their generations, in their nations, and by these were the nations divided in the earth after the flood. Chapter 11. And the whole earth was of one language and of one speech. And it came to pass, as they journeyed from the east, that they found a plain in the land of Shinar, and they dwelt there. And they said one to another, Go to, let us make brick, and burn them thoroughly. And they had brick for stone, and slime had they for mortar. And they said, Go to, let us build us a city and a tower, whose top may reach unto heaven, and let us make us a name, lest we be scattered abroad upon the face of the whole earth. And the Lord came down to see the city and the tower which the children of men builded. And the Lord said, Behold, the people is one, and they have all one language, and this they begin to do. And now nothing will be restrained from them which they have imagined to do. Go to, let us go down, and there confound their language, that they may not understand one another's speech. So the Lord scattered them abroad from thence upon the face of all the earth, and they left off to build the city. Therefore is the name of it called Babel, because the Lord did there confound the language of all the earth. And from thence did the Lord scatter them abroad upon the face of all the earth.
Language families are important aspects when trying to determine the descendants of Shem, Ham, and Japheth, specifically proto-language families, since language was used to separate these people. We can combine our understanding with Y DNA haplogroups and language. After all, people create languages and people carry haplogroups. Therefore, an ethnic group who first spoke their native language are likely all genetically related. With that being said, let's begin. I believe haplogroups formed after the flood because humans prior to the flood were genetically pure and had no mutations. But after the flood, the pre-flood world environment changed, and therefore humans changed as well, causing genetic mutations to occur. However, some people could make the argument that genetic mutations started to occur close to the time period of which the flood was going to occur, and Shem, Ham, and Japheth themselves could have had a genetic mutation as well.
And what happened is when God created Adam and he sinned, he fell, that errors came into the code. The master programmer endowed us with perfect code, and only at the fall of man did it become corrupted.
Number one, they would have a pure gene pool, no defective chromosomes. Today, the average person has about 4,000 defective genes. A gene, in case you don't know, is if you took a ladder, an extension ladder from here to Chicago, about 900 miles, and you had somebody stand on the other end while you started twisting it here. You twisted and twisted and twisted and twisted. By the time it gets all twisted up from here to Chicago, that would be roughly what a chromosome looks like. Each cell in your body has 46 of those little chromosomes. The rung of the ladder would be a gene. So the illustration people use is a twisted ladder. Pretty tough to draw 2D, what a 3D ladder would look like twisted. But each of these things across here is a rung of the ladder. Each of those is called a gene. The whole thing is called a chromosome, or a DNA strand, deoxyribonucleic acid. The same thing as a chromosome, deoxyribonucleic acid. And this thing right across here is one gene, G-E-N-E. Now, when you get regenerated in Christ, you're being regened, totally new person, new genes. That's where the word regenerate comes from that word gene. Now that'll preach too. But when during conception for the baby, half of this chromosome comes from the mother and half comes from the father. The problem is it splits down the middle. Now let's take a ladder that is twisted from here to Chicago. We're going to split it all the way down the middle. It's going to unwind itself, and it's going to get the other half of the ladder from the mother or father, depending which the case may be, and it's going to wind itself back together, and all those genes are going to line up. Absolutely phenomenal how this can happen. And that's one chromosome. You got 46 that are doing this. 46 chromosomes in every cell of your body. So radiation from the sun is one of the things that causes damage to these genes. You often see people at the hospital when they're going to get an x-ray, they will put a lead apron over where the ovaries are on a woman, for instance, because they know the x-ray could damage the chromosomes, could damage the genes for the next generation. Many things can damage this, and so they try to be very sensitive to not create damage to this. But one of the reasons they lived to be 900 before the flood was they had a pure gene pool, no genetic load. You've got about 4,000 of these genes in your gene code that are defective. Sort of like a computer program where something's wrong. Now, if you marry closer than a first cousin, there's a high probability that you both have a similar gene code, and so you might both have the same defective genes. Now you're asking for trouble as far as deformed children. It's against the law in all but 3 states to marry closer than a first cousin. You have to marry second cousin or more farther away in all but 3 states in America. I won't tell you which the 3 states are, but drive through them and you'll be able to tell. The people anyway, the people were living long before the flood. They had no defective chromosomes in the original creation, number one. Number two, they had a canopy of water to protect them, to filter out the radiation from the sun. Number three, they're eating a perfect diet. They're also eating food that has been unaffected by any of this harmful stuff. Plus, they're eating food grown in soil that is not depleted at all, mineral-rich soil. I think in the original creation, they had perfect food supply, perfect atmosphere to breathe, no genetic load. They had probably several reasons why they were living to be 900 years old.
So the chemical code built on A, G, T, and C becomes corrupted as a result of the shuffling of the order of those bits, or a loss of one or more bits. And Adam, as the father of the race and a direct creation of God, had a perfect code. There was no loss of data or corruption of the data in any way. And it was when he fell that those things in some way got scrambled, or they lost some data, or something happened there where there was a loss of information. And John Sanford puts it this way. He says the extinction of the human genome appears to be just as certain and deterministic as the extinction of stars, the death of organisms, and the heat death of the universe. So he's discovered, and others have as well, that the entire human race is about 50% dead. We're on our way to extinction as a race, and because there are more and more mutations that continue to get into our genome. You really are more of a mutant than your parents, in case you're wondering your children are more mutant than you are. So it's not just some feeling that you had, but it's true. They really are more mutant. But we're all mutant. We're all mutating, and we're losing information. Well, during normal conception, not looking at the miraculous conception at this point. During reproduction, the maternal and paternal gametes, that is the haploid cells, fuse at conception to produce a zygote, which will turn into a fetus and eventually into an adult human being. And when two gametes unite during fertilization, each contributes its haploid set of chromosomes to the new individual, restoring the diploid number. So we can essentially look at it this way, is that Mary provided one half, and the Holy Spirit provided the other half, thus making the diploid number, which of course became the 46 unique chromosomes, or the 23 base pairs needed for the body of Jesus Christ. And that's the amazing thing, is that you have the terrestrial Mary with the celestial Holy Spirit combining to make the body of Jesus Christ. It's truly amazing. And now that we're in the genetic age, we can just catch a glimpse of what that miracle entailed. Now there's something else that happened that is quite astounding. We know that sin came through Adam, and as a result of sin, death spread to everything because of his sin. And we refer to this as the original sin. Well, it turns out that a son will inherit an identical copy of his father's Y chromosome, and this copy is also essentially identical to the Y chromosomes carried by all his paternal forefathers across the generations. We also read a male shares the same Y chromosome with his father, paternal grandfather, paternal great-grandfather, and so on. So, according to Neil Bradman and Mark Thomas, who are, incidentally, committed evolutionists, they don't believe in God or anything of this stuff, but they actually rewrote Genesis chapter 5 in modern terms. They say, Genesis chapter 5 records the generations of Adam. Adam begot Seth, Seth begot Enosh, etc., down to Noah of the flood. Translated into modern genetic terms, the account could read, Adam passed a copy of his Y chromosome to Seth, Seth passed a copy of his Y chromosome to Enosh, Enosh passed a copy of his Y chromosome to Kenan, and so on until Noah was born carrying a copy of Adam's Y chromosome. Now this becomes very fascinating when we consider that, still according to these guys, they say that the Y chromosome may in fact be a record of an event in the life of our original father. Bradman and Thomas suggest that the Y chromosome contains a record of an event in the life of the man who passed on the current Y chromosome, which they say had no effect on the life of the man in whom the change occurred, nor indeed on the life of his descendants. However, is it possible that the recorded event is not something that had little or no effect, but is in some way the record of the genetic fall of our first father? And obviously I argue that that is indeed the case.
The early descendants of Shem, Ham, and Japheth likely are the first ones to form haplogroups, but like I said, you could also make the argument that Shem, Ham, and Japheth themselves would have been the first to have haplogroups. It depends on your model. Nevertheless, I somewhat reject the phylogenetic tree, but I do understand the concept and its importance. Basically, I agree with it to a certain extent, such as I agree that all humans today have a common ancestor, and that haplogroups can be used to trace back to that common ancestor. But I believe post-flood humanity traced their lineage back to Noah, their most common recent ancestor. I believe haplogroups form 3 ancestral nodes that can be used to trace back to Shem, Ham, and Japheth. PhD Dr. Nathaniel Jansen, who studied at Harvard University, has data illustrating 3 ancestral nodes that represent the wives of Shem, Ham, and Japheth. According to the article at Institute for Creation Research titled DNA Trends Confirm Noah's Family, it reads, when research biologist Dr. Nathaniel Jansen plotted hundreds of human mitochondrial DNA sequences onto a tree diagram, the project revealed an obvious pattern. The mtDNA stemmed from 3 central trunks or nodes instead of just 1. 3 trends in Jansen's data suggest that the wives of Noah's sons, Shem, Ham, and Japheth, best explains this finding. Jansen first downloaded mtDNA sequences taken from all major human groups. He then used standard software that arranges the most similar sequences closest together. The result is a tree-like diagram depicting lines of ancestry. Jansen's data show that the human mtDNA tree has 3 nodes. Thus, everyone alive today carries one of the 3 unique ancestral maternal sequences. This fits Genesis' claim that all humans who exist today descend from one of the wives of Noah's sons. We find the second trend in the number of DNA differences between the three central nodes. At today's mtDNA mutation rate, 2 to 8 nucleotide differences would have accumulated in the 9th generation between Adam and Noah, and the difference between the three central nodes also shows 8 DNA differences. And according to Answers Research Journal, titled On the Origin of Human Maternal DNA Differences, New Generation Time, data both suggest a unique young Earth creation model and challenge the evolutionary out of Africa model. It reads, in theory, among these three wives, Africans and Asians may have traced their ancestry back to different women. Consistent with this hypothesis, the highly divergent San and Taiwanese Aborigine individuals traced their ancestry back to different mtDNA nodes. Thus, not only did the San and Taiwanese Aborigine not share a common ancestor after the Tower of Babel incident, they also appeared to have different maternal ancestors at the time of the flood. Human mtDNA tree for 369 individuals of varying ethnicities. The 369 human mtDNA sequences clustered into three major nodes, which were indicated by green block arrow. One of the nodes spawned almost exclusively sub-Saharan African individuals, whereas the other two nodes gave rise predominantly to non-African and North African individuals. The position of the 6 South African individuals analyzed in Table 4 were indicated with blue block arrows and underlines. The position of the individuals from the primarily African node with shorter branch lengths were indicated by gold arrows. The most divergent pair of individuals was between the San and Taiwanese Aborigine individuals, noted by the red block arrows and underlines. Just like Dr. Nathaniel Jansen mapped out 3 ancestral nodes for the wives of Shem, Ham, and Japheth, I believe the same thing exists for the male lineage of Shem, Ham, and Japheth. And I also believe in an out of Asia theory for this new branch of humanity because Noah's Ark rested in Asia, in the Middle East. And there's a 2020 paper titled The Reversal of Human Phylogeny. Homo left Africa as Erectus came back as Sapiens sapien, which states that the most common ancestral lineage Y-DNA Adam is suggested to have originated somewhere in the Middle East or the Iranian Plateau. This would mean Africans and Eurasians both came from West Asia and separated from West Asia to Africa and other parts of the world, fitting with the idea of Shem, Ham, and Japheth descendants all being generally around the same area with each other before dispersing. My model for the breakdown of Shem, Ham, and Japheth is this. Ham would be represented by haplogroup A and B. Japheth would be represented by haplogroup CF, and Shem would be represented by haplogroup DE. These 3 Y-DNA haplogroups would represent the ancestral nodes for all men today, post-flood, going back to Shem, Ham, and Japheth. In the future, I'll do a thorough breakdown of the haplogroups of Shem, Ham, and Japheth, but for now, I'm going to do a very quick breakdown for the various sons of Noah. So let's begin with Ham. Ham, son of Noah. According to the scriptures, it's generally believed that Ham's descendants settled Africa. Therefore, I believe Ham is haplogroup A and B. Bears of haplogroup A have been found in southern Africa's hunter-gatherer inhabited areas, especially among the San people. In addition, the most basal mitochondrial DNA, LO, lineages are also largely restricted to the sun. However, the A lineages in southern Africa are subclades of A lineages found in other parts of Africa, suggesting that A sub-haplogroups arrived in southern Africa from elsewhere. The two most basal lineages of haplogroup A, A0, and A1 have been detected in West Africa, Northwest Africa, and Central Africa. Kornicki et al. 2011 suggests that these lineages may have emerged somewhere in between Central and Northwest Africa. Scornozzi et al. 2012 also support the hypothesis of an origin in the northwestern quadrant of the African continent for A1b haplogroup. Haplogroup A1b1b2 has been found among ancient fossils excavated at Belto Bay of KwaZulu-Natal, South Africa, which have been dated to around 2194 through 1831 BP. By the definition of haplogroup A as non-BT, it is also completely restricted to Africa, though a very small handful of bearers have been reported in Europe and Western Asia. The clade achieves its highest frequency among the Bushman hunter-gatherer populations of southern Africa, followed closely by many Nilotic groups in eastern Africa. However, haplogroup A's oldest subclades are exclusively found in central northwestern Africa, where it is believed to have originated. Haplogroup A is found at low to moderate frequencies in the Horn of Africa. The clade is observed at highest frequencies among the 41% of a sample of the Beta Israel, occurring among 41% of one sample from this population. Elsewhere in the region, haplogroup A has also been reported in 14.6% of the Amhara sample, 10.3% of the Oromo sample, and 13.6% of another sample from Ethiopia. In the Upper Nile, haplogroup A3b2-M13 is common among southern Sudanese at 53%, especially the Dinka Sudanese at 61.5%. Haplogroup A3b2-M13 also has been observed in another sample of the southern Sudanese population at a frequency of 45%. Further down around the Nile Valley, the subclade A3b2 has also been observed at very low frequencies in a sample of Egyptian males at 3%. One 2005 study has found haplogroup a sample of various Khoisan-speaking tribes with frequencies ranging from 10% to 70%. This particular haplogroup was not found in a sample of the Hanza from Tanzania, a population somewhat proposed as remnant of a late Stone Age Khoisan population. In Asia, haplogroup A has been observed at low frequencies in Asia Minor, which is Turkey, and the Middle East among Aegean Turks, Palestinians, Jordanians, and Yemenis. A3A2, A-M13, has been observed at very low frequencies in some Mediterranean islands. Without testing for any subclade, haplogroup A has been found in a sample of Greeks from Mytilene, Aegean island of Lesbos, and in samples of Portuguese from southern Portugal, central Portugal, and Madeira. The authors of one study have reported finding what appears to be haplogroup A in 3.1% of a sample of Cretans. The clade A1b1b contains the most populous branches of haplogroup A and is mainly found in eastern and southern Africa. The subclade A1b1b2b is primarily distributed among Nilotic populations in East Africa and northern Cameroon. It is different from the A subclades that are found in the Khoisan samples and only remotely related to them. It is actually only one of many subclades within haplogroup A. This finding suggests an ancient divergence. In Sudan, haplogroup A-M13 has been found in 52.8% of southern Sudanese, 46.4% of the Nuba of central Sudan, 27.8% of western Sudanese, 12.5% of local Hausa people, 2.3% of northern Sudanese. In Ethiopia, one study has reported finding haplogroup A-M13 in 14.6% of a sample of Amhara and 10.3% of a sample of Oromo. Haplogroup A-M13 also has been observed occasionally outside of Central and Eastern Africa as the Aegean region of Turkey, 6.7%, Yemenite Jews at 5%, Egypt at 2.7%, Palestinian Arabs at 1.4%, Sardinia at 1.3%, the capital of Jordan, Amman, at 1%, and Oman at 0.8%. Haplogroup A-M13 has been found among 3 Neolithic period fossils excavated from the Kadrutika site in Sudan. Haplogroup B is common in parts of Africa, especially the tropical forests of West Central Africa. It was the ancestral haplogroup of not only modern pygmies like the Baka and Mbatwi, but also the Hadza from Tanzania, who often have been considered in large part because of some typological features of their language to be a remnant of Khoisan people in East Africa. According to one study of the Y-DNA of populations in Sudan, haplogroup B-M60 is found in approximately 30% of southern Sudanese, 16% of local Hausa people, 14% of the Nuba of central Sudan, 3.7% of northern Sudanese, but only among Copts and Nubians, and 2.2% of western Sudanese. According to another study, haplogroup B is found in approximately 15% of Sudanese males, including 12.5%. In Madagascar, haplogroup B-M60 has been found in approximately 9% of Malagasy males, including 6%. Family tree DNA shows a significant number of persons of haplogroup B-M60 claiming origins from the Arabian Peninsula, dominantly Saudi Arabia, but also in Kuwait, Bahrain, Yemen, Qatar, Iraq, United Arab Emirates, and Oman. In Hormozgan province in Iran, haplogroup B-M60 has been found in 8.2% of a sample of 49 Kashmir people, and in 2.3% of a sample of 131 Bendari people. In Afghanistan, haplogroup B-M60 has been found in 5.1% of a sample of Huzarus males. Haplogroup B-M182 has been found in 6% of the sample of Mbatwi males from the Democratic Republic of the Congo, 6% of Bakola males from southern Cameroon, 6% of a sample of Dama males from Namibia, and 3% of a sample of Baika males from Central African Republic. The vast majority of family tree DNA participants in haplogroup B-M60 test positive for B-M182, with 3/4 of those participants claiming countries of the Arabian Peninsula as their ancestral land of origin, attesting to its presence in that area also. Haplogroup B-M112 has been found mainly among pygmy populations in Central Africa, northern Khoisan populations in Southern Africa, and the Hadza in East Africa. Specifically, haplogroup B2B has been observed in 67% of a population of Baka from Central African Republic, 52% or 51% of a sample of Hadza from Tanzania, 48% of a sample of Baka from Central African Republic, 43% of a sample of Mbatwi from the Democratic Republic of the Congo, 31% of a sample of San from Namibia, 28% of a sample of the Northern Khoisan speaking peoples, 25% of a sample of Barunj from Tanzania, 14% of sample of Tutsi from Rwanda, 13% of a sample of Sinawi from Tanzania, 9% of a sample of Kong from Namibia, 5% of a sample of Tortur from Tanzania, 5% of a sample of Warak from Tanzania, 3% of a sample of Zulu from South Africa, 3% of a sample of Bakola from southern Cameroon, 3% of a sample of Datong from Tanzania, 3% of a sample of Malagasy, 1.4% of a sample of Hutu from Rwanda, 1.4% of a sample from Qatar, and 1.3% of a sample from Saudi Arabia. The signature nilotic paternal marker haplogroup A3b2 was observed in 27% of the Maasai, 22% of the Ulur, and 11% of the Luyo. Haplogroup B is another characteristically nilotic paternal marker. It was found in 22% of the Luyo samples, 8% of the Maasai, and 50% of the Nuer people. The y-DNA of populations in the Sudan region were studied with various local nilotic groups included for comparison. The signature nilotic A and B clades were the most common paternal lineages among the Nilo-Saharan speakers, except those inhabiting Western Sudan. There, a prominent North African influence is noted. Haplogroup A was observed among 62% of the Dinka, 53% of Shilluk, 46.4% of the Nuba, 33.3% of the Nuer, 31.3% of the Fur, and 18.8% of the Masalit. Haplogroup B was found in 50% of the Nuer, 26.7% of the Shilluk, 23% of Dinka, 14.3% of Nuba, 3.1% of Fur, and 3.1% of Masalit. According to the paper titled Y Chromosome Variation Among Sudanese Restricted Gene Flow Concordance with Language, Geography, and History, it reads, haplogroups A-M13 and B-M60 are present at high frequencies in Nilo-Saharan groups except Nubians with low frequencies and Afro-Asiatic groups, although no table frequency of B-M60 were found in Hausa 15.6% and Copts at 15.2%. And according to the paper titled New Binary Polymorphisms Reshape and Increase Resolution of the Human Y Chromosomal Haplogroup Tree, it reads, this subclade, one of the most basal of the entire Y chromosome tree, is also completely restricted to the African continent. Haplogroup A chromosomes are most frequent in Khoisan, Ethiopian, and Sudanese populations. Like haplogroup A, the B clade is almost entirely restricted to sub-Saharan Africa. B chromosomes are found at their highest frequency among pygmies, with some lineages being virtually restricted to these ethnic groups. And according to the paper titled Signatures of the Pre-Agricultural Peopling Processes in Sub-Saharan Africa has revealed by phylogeography of early Y chromosome lineages, and it reads, the study of Y chromosome variation has helped reconstruct demographic events associating with the spread of language, agriculture, and pastoralism in sub-Saharan Africa, but little attention has been given to the early history of the continent. In order to overcome this lack of knowledge, we carried out a phylogeographic analysis of haplogroups A and B in a broad data set of sub-Saharan African populations. These two lineages were particularly suitable for branches of the Y chromosome genealogy. Their distribution is almost exclusively restricted to sub-Saharan Africa, where their frequencies peak at 65% in groups of foragers. The combined high-resolution single nucleotide polymorphism analysis with short tandem repeats variations of their subclades reveals strong geographic and population structure for both haplogroups. This has allowed us to identify significant lineages related to regional pre-agricultural demographics in different areas of sub-Saharan Africa. In addition, we observed signatures of relatively recent contact both among pygmies and between them, the Khoisan speakers, groups from southern Africa, thus contributing to the understanding of the complex evolutionary relationship among African hunter-gatherers. Haplogroup A is rarely found in North, West, and Central Africa, whereas it is more frequent in the eastern and southern parts of the continent. Rare in both northern and western Africa, the distribution of haplogroup B in the rest of the continent can be described by that of its two main subclades, B2A and B2B. The former appears to be associated with food-producing communities and populations in contact with them, as also previously observed, and it is present at low frequencies in all sub-Saharan areas. In contrast, B2B is mostly present in foraging communities in Eastern and Central Africa. The different geographic distributions of haplogroups A and B2B are mirrored at the population level. Little or no haplogroup A is present in pygmies and Eastern African Khoisan speakers, whereas B2B is commonly found in these populations. On the other hand, haplogroup A is more frequent than B among Southern African Khoisan speakers, less than 40%, with haplogroup B2B representing less than 16% of the Y chromosome types present in these populations. Of the A subclades, A1 is found only in Western and Central Africa, whereas A3B1 and A3B2 are Southern and Central/Eastern Africa specific, respectively. Haplogroup A2 is mostly represented by sub-Saharan samples, with only a few Central African haplotypes. Similarly, B2B1/B2B4A and B2B2 are geographically restricted to Southern and Eastern Africa, respectively, whereas B2B3, B2B4B, and B2B4 are specific to Central Africa, albeit with a few B2B4 sub-Saharan haplotypes. In fact, whereas B2B3, B2B4B, and B2B4 are almost exclusively found among Western pygmies, B2B and B2B1-B2B4A are found only in Eastern pygmies and sub-Saharan Khoisan speakers, respectively. Similarly, the majority of the A3B1 and A2 types are found among sub-Saharan Khoisan speakers, with haplogroup A2 also present in Western pygmies. Pygmies and sub-Saharan Khoisan speakers also share evolutionary closely related lineages within the B2B4 clade. We dated the Eastern and Western pygmy separation using the divergence between the B2B2 and B2B4B/B2B3 clades. These estimates span similar time intervals and suggest a separation time of 10,000 through 15,000 years ago, broadly overlapping with the generally more ancient estimates provided by mtDNA and autosomal data. In particular, the youngest date suggested by B2B2 versus B2B3 might indicate post-late glacial maximum. We identified evolutionary links between western pygmies and sand in both A and B clades, developing the initial findings presented in Wood et al. 2005. Haplogroup A2, found among sub-Saharan Khoisan speakers at 25 through 45%, was detected for the first time in the present work of non-interval frequency 5% among the Baka pygmies from Cameroon and Gabon. On the other hand, B2B4 was present at 6% through 7% among Khoisan speakers, but reached 45 through 67% in both Baka and Bainka pygmies. We dated the TMRCA among the Western Pygmy and San specific subclades of these two haplogroups to between 3,000 and 4,000 years ago. Haplogroup A in Western Africa is represented by the A1A lineage. The variation within this subclade dates back to 10.5 KYA and to 8 KYA, when only Western African haplotypes are considered, which is in agreement with the archaeological and linguistic evidence related to the peopling of this region. Haplogroup B2B4 chromosomes were present in the Mozambican samples, a lineage that is mainly shared with the Baka pygmies from Cameroon. This subclade A3B2 is present at high frequencies in East African populations, in particular among Nilo-Saharan speakers. Based on the analysis of this lineage in Uganda, proposed its association with the linguistic phylum, our estimates of A3B2 antiquity do not refute this hypothesis, as they are broadly in agreement with the interval date for the spread of Nilo-Saharan phylum, approximately between 12,000 and 18,000 years ago. To put it simply, Ham's descendants would be represented by haplogroups A and B. Haplogroup A and B is Paleo-African or True African. Ethnic groups with haplogroup A and B would be Hamites such as Pygmies, Khoisan, Nilo-Saharans, Nilotic people, Sudanese, Upper Egyptians, Coptics, and Ethiopians. And just to visualize the tree of haplogroups A and B, here is a paper titled Molecular Dissection of the Basal Clades in the Human Y Chromosome Phylogenetic Tree. And there, as you can see, haplogroups A and B. Those subclades would represent the descendants of Ham. Haplogroups A and B. Haplogroup A and B. Haplogroup A is the NRY macrohaplogroup, from which all modern paternal haplogroups descend. It is sparsely distributed in Africa, being concentrated among Khoisan populations in the southwest and Nilotic populations towards the northeast in the Nile Valley. Here is a distribution map of haplogroup A. As we can see, highest frequency is among Nilotic and Khoisan peoples, and it is highly distributed in the Sudan and in South Africa, but you can also find it in all parts of Africa at low frequencies. And here is a map of haplogroup B, which is also distributed across Africa, and you can find it in higher frequencies among the Pygmies, but it is evenly distributed all around the continent. But haplogroup A and B in ancient times was all across the continent, even in northern Africa, and haplogroup A and B seems to be the best choice for Ham, since it is a mostly African restricted haplogroup seen among the oldest African populations such as Nilots and Pygmies and Khoisans, etc. Even in Western Africa, the oldest samples we find is haplogroup A and B. For that information, check out the paper titled Ancient West African Foragers in the Context of African Population History. That paper details haplogroups A and B being the oldest in Western Africa. Since Ham is haplogroup A and B in my model, haplogroup A and B is seen highest among Nilo-Saharan, Pygmies, and Khoisanes. Therefore, the actual language of Ham would be those languages. The African Pygmies are a group of ethnicities native to Central Africa, most of the Congo Basin, traditionally subsisting on a forager and hunter-gatherer lifestyle. They are divided into three roughly geographic groups, such as Western Pygmies, Eastern Pygmies, and Central and Southern Pygmies, with their respective languages. And just to give a slight description of who the Pygmy people are, the type of African Pygmies living in Central Africa shows the strongest adaptation to tropical rainforest conditions of all humans. Mbatuids belong to the shortest humans. The males hardly surplus 140 centimeters. Very short-legged, athletic. Noses are the widest in the world, often wider than the mouth. Features: overall infantile. Hairiness shorter than the savanna groups. Lips thicker. Skin fairer. Faces wide and peppercorn. Hair ranged from reddish blonde to black. The types diverge from others some 60,000 years ago and developed through hunter-gatherer lifestyles in the forest. East Mbatuids show the most pronounced features. Their stature is shorter, face wider, nasal root lower, and pronaphany weaker than the Western Mbatuids. The similar Twa group shows stronger Congoid influence. Most Mbatuid tribes today suffer from war, discrimination, and loss of culture. The Khoisan languages, a group of African languages originally classified together by Joseph Greenberg. Khoisan languages share click cognates and do not belong to other African language families. And here is a tree of Khoisan languages and ethnic groups, the people who speak those different sub-branches of the language. Now to give a brief description of the Khoisan peoples. Special group, typical for the old agricultural population of southern Africa, with many unique features adapted to dry climate. Very ancient and distinct. Stature is rather short and body are lean. Women show steatopygia. The skin is yellowish light brown, often wrinkled, peppercorned hair common. Nose very wide. Eyes fold are frequent. The skull is long and low with little pronathism. Compared to the similar Sanids, Khoisades are taller, less infantile, faces larger, chins more pointed, heads larger. The convex upper lip is absent. Was more widespread across southern Africa until Bantu and European colonization almost wiped it out. Besides the Khoinoid proper, distinct relationships to East African types may exist. Sahanwi, Hadza, as well as North African types. Paleo-Saharan. The almost extinct Strandhopper type seems to be the ancestral form. The Nilo-Saharan languages are a prominent family of African languages spoken by some 50 through 60 million people, mainly in the upper parts of the Shari and Nile rivers, including historic Nubia, north of where the two tributaries of the Nile meet. The languages extend through 17 nations in the northern half of Africa, from Algeria to Benin in West Africa, from Libya to the Democratic Republic of the Congo in the century, and from Egypt to Tanzania in East Africa. And here is a visualization of the Nilo-Saharan languages and its subgroups and the people who speak them. And now just a quick description of the Nilotic people. specialized African type native to the swamps and savannas of the Upper Nile region in South Sudan and adjacent countries. With their black skin, they belong to the darkest people on earth. Their thin bodies are the tallest in all indigenous human groups worldwide who are still alive. Their legs longer than any other known group. Only poverty and malnourishment keep individuals below their genetic height potential. Heads long, facial features coarse, with a rather wide nose, full lips, and kinky hair. Pronathism weak. Nilots seem to be a relatively old group that might have extended further east into Ethiopia during prehistoric periods. The most typical variety is the Dinka, native to swamp regions. A similar savanna counterpart is the Shuluku. South Nilotic shows less pronounced features, possibly due to admixture, and Chad, the admixed West Nilotic Shari type, is found. A very ancient variety still exists between Sudan and Ethiopia, the pre-Nilot. Since I believe Ham is haplogroup A and B, his descendants today range in many different skin tones, from jet black of the Nilots to the dark brown Pygmies to the medium brown Ethiopians and the light brown Khoisan and the tanned skinned Copts. These are the faces of Ham. So, in summary and conclusion, Ham is haplogroup A and B. Haplogroup A and B is Paleo-African. Haplogroup A and B is seen highest among Nilo-Saharan, Pygmies, and Khoisans. The language of Ham would be Nilo-Saharan, Pygmy, and Khoisan. Other African populations with haplogroup A and B are Coptic, Beta Israel, Bantu, Nilotic, Sudanese, etc. And haplogroup A and B has also extended into parts of the Middle East, seen among peoples such as the Saudis and Yemenis, Palestinians, and various others. And we also see that haplogroups A and B is in the Mediterranean, such as the Cretans in Greece. And the Hamites in skin tone range from jet black to dark brown to medium brown and to light brown. In the future, I'll do a more in-depth video on each son of Ham and his grandsons. But for now, this is what I have to say about Ham and his haplogroup and his descendants. And this is a good basic understanding when it comes to Ham. Now let's turn our attention to Japheth. It's generally believed that Japheth's descendants settled in Eurasia. Japheth would be the father of most Eurasian peoples, such as Indo-Europeans, Caucasians, Turkic tribes, East Asians, Native Americans, Oceanic populations, and various other Eurasian peoples. According to Amazing Bible Timeline with World History titled Japheth, Son of Noah, it reads, Japheth is usually thought to be the father of the Indo-Europeans and the father of the Japhetic race, equating the Japhetic nations to Europeans. He is known to be the ancestor of all Indo-European nations. The majority of his descendants are situated in northwestern regions like Anatolia and Aegea. However, there is also an assumption that Japheth is the father of the Asian or Mongoloid people. After what happened to the Tower of Babel, the Japhetic race went to the east of Europe and north of Asia to propagate. The Bible phrases that states they migrated to the Isles of the Gentiles is generally thought to be the Greek Isles, but some consider that it refers to the islands in Asia and the Pacific Islands. His sons and their people started to multiply in the mountains of Tartarus and Amanus. Then they dwelt to the lands of presently known as the Mediterranean, Russia, Asia, and the Americas. Therefore, I believe that Japheth is haplogroup CF, which is the most commonly held paternal haplogroup in most Eurasian populations, like Indo-Europeans, Caucasians, Central Asian Turks and Mongolians, East Asian Native Americans, Oceanic populations, and Northern Eurasian populations, etc. Haplogroup CF, also known as CF-P143, is a human Y chromosome DNA haplogroup. This paternal lineage is defined by the SNP P143. The clade's existence and distribution are inferred from the fact that haplogroups descend from CF include most human male lineages in Eurasia, Oceania, and the Americas. CF is an independent descendant of the CT and is the sibling of haplogroup DE YAP. It is the independent ancestor of both haplogroup C and haplogroup F. Haplogroup F, also known as F-M89, is a very common Y chromosome haplogroup. The clade and its subclades constitute over 90% of paternal lineages outside of Africa. The vast majority of individual males with F-M89 falls into its distinct descendant haplogroup GHIJK. Haplogroup GHIJK branches subsequently into two different descendants, G and HIJK. HIJK in turn splits into H and IJK. The descendants of the haplogroup IJK include the clades IJK, and ultimately several major haplogroups descend from haplogroup K, namely haplogroups M, N, O, P, Q, R, S, L, and T. According to the paper titled New Binary Polymorphism, Reshape and Increase Resolution of the Human Y Chromosomal Haplogroup Tree, and it reads, haplogroup C still has not been detected in sub-Saharan African populations, suggesting an Asian origin after anatomically modern humans migrated out of Africa. The ancestral power group of haplogroup C, as well as many downstream lineages, are commonly found among Asian, Australian, and Oceanic populations. Recently, a sublineage of haplogroup C was found to be restricted to Native American populations, while branch C4 or C-M347 was observed only in Australia. So just to name a few locations where haplogroup C was found, haplogroup C was found in Asia, Oceania, North America, Japan in low frequencies in Europe, Armenians in Algeria. It's also been found in southern South Asia, southwestern Asia, and northern China. It's been found also in low frequencies in Saudi Arabia and Iraq. Also been found in Indonesia, New Guinea, Melanesia, Polynesia. It's found among indigenous peoples of Australia, and it's found all throughout Eurasia, North America, Mongolians, Tuscan people, Paleo-Siberians, etc. To simply put it, haplogroup C found in Oceania, North, Central, East Asia, North America, and minor presence in South America, Southeast Asia, South Asia, West Asia, and Europe. Haplogroup G. The G clade is not widely distributed, being present mostly in the Middle East, the Mediterranean, and the Caucasus. And just to give a brief description of haplogroup G, It is found in many ethnic groups in Eurasia, most common in the Caucasus, Iran, Anatolia, and the Levant, found in almost all European countries, but most common in southeastern Romania, Greece, Italy, Spain, Portugal, etc., with highest concentrations on some Mediterranean islands, uncommon in northern Europe. G-M201 is also found in small numbers in northwest China and India, Pakistan, Sri Lanka, Malaysia, and North Africa, etc. To simply put it, haplogroup G is present among many ethnic groups in Eurasia, usually at low frequencies, most common in the Caucasus, the Iranian Plateau, and Anatolia. In Europe, mainly in Greece, Italy, Iberia, and other parts. Haplogroup H. The geographic distribution of haplogroup H is almost entirely restricted to the Indian subcontinent. Its subclades are also found in low frequencies in Iran, Central Asia, across the Middle East, and the Arabian Peninsula. However, H2 is present in Europe since the Neolithic, and H1A1 spread westward in the medieval era with the migration of the Roma people, which are gypsies. H1A, India, Sri Lanka, Nepal, Pakistan, Iran, Central Asia. H1B, found in Burmese individuals. H3, India, Sri Lanka, Pakistan, Bahrain, Qatar. H2, formerly known as haplogroup F3, found with low frequency in Europe and Western Asia. To put it bluntly, haplogroup H is typically seen in India, Sri Lanka, Nepal, Pakistan, Iran, and Central Asia, as well as in some parts of Europe with the Gypsies or the Roma people, and in some parts of the Middle East and East Asia. Haplogroup I. Clade I represents one of two major European Y chromosome haplogroups, with haplogroup R being the other major group. Unlike R, clade I is widespread in Europe and is virtually absent elsewhere. Subclade I1 is found mostly in Northern Europe, while subclade I2 is the most frequent haplogroup in Eastern Europe and the Balkans. Haplogroup I is found mainly in Europe and the Caucasus. I1, Nordid/Nordic Euripids, found mainly in Northern Europe. Haplogroup I2, Dreniid or Drenic Euripids, found mainly in Balkans, Southeast Europe, and Sardinia, save for I2b1, which is found at a moderate frequency in Western, Central, and Northern Europe. So to put it basically, haplogroup I1 is widespread in Europe and is found in some parts of the Middle East, but is virtually absent elsewhere. And haplogroup I1 is typically in Northern Europe, dominated in Scandinavia, and haplogroup I12 is basically seen in Central and Southeast Europe, Sardinia, and the Balkans. Haplogroup J. Haplogroup J's lineages are found at high frequencies in the Middle East, North Africa, Europe, Central Asia, Pakistan, and India, with haplogroup J-M172 being the most common J haplogroup in Europe, while haplogroup J-M267 predominates in Middle East, North Africa, and Ethiopia. Basically, haplogroup J is seen in the Middle East, Turkey, Caucasus, Italy, Greece, the Balkans, and North Africa, etc. Haplogroup L. The majority of L haplogroups are found on the Indian subcontinent. Haplogroup L chromosomes are also present in the Middle East, Central Asia, Northern Africa, and Europe along the Mediterranean coast. To simply put it, haplogroup L is found in South Asia, Central Asia, Southwest Asia, and the Mediterranean. Haplogroup T. Clade T was observed at low frequencies in the Middle East, Africa, and Europe. Haplogroup T is found at high levels in the Horn of Africa, mainly Cushitic-speaking peoples, parts of South Asia, the Middle East, and the Mediterranean. T-M184 is also found in significant minorities of Egyptians, Omani, Sephardi Jews, etc. It is also found at low frequencies in other parts of the Mediterranean and South Asia. To put it simply, haplogroup T is found in North Africa, Horn of Africa, Southwest Asia, the Mediterranean, South Asia, formerly known as haplogroup K2. Haplogroup N. Haplogroup N is mainly found in northern Eurasia and is absent or marginally present in other regions of the globe. Haplogroup N is found through northern Eurasia, especially among speakers of the Ulric languages. Haplogroup N possibly originated in Eastern Asia and spread both northward and westward into Siberia, being the most common group found in some Yorick-speaking peoples. So, to put it simply, haplogroup N is found in northernmost of Eurasia. Haplogroup O. Haplogroup O is the major haplogroup in East Asia. Haplogroup O chromosomes are also found in Central Asia and Oceania at moderate or low frequencies. Haplogroup O is found with its highest frequency in East Asia and Southeast Asia, with lower frequencies in the South Pacific, Central Asia, South Asia, and islands in the Indian Ocean. Haplogroup O1, haplogroup O1A, found in eastern, central, and southern mainland China, Taiwan, and Southeast Asia. Haplogroup O1B1, found in Japan, southern China, Taiwan, Southeast Asia, and the Indian subcontinent. Haplogroup O1B2, found in Japan and Southeast Asia and other regions, and haplogroup O, found throughout East Asia, Southeast Asia, and Australasia, including Polynesia. Just to put it simply, haplogroup O is found in East Asia, Southeast Asia, the Pacific Islands, South Asia, and Central Asia. Haplogroup K. Instead, haplogroup K is characterized by the derived state at 4 sites, the ancestral state at the mutation that define the L, M, N, O, P, S, and T lineages. Haplogroup K1 was found at low frequencies in India/Pakistan. Lineages K2, K3, and K4 are found in Oceania, Indonesia, and/or Australia. K2 was renamed as a new haplogroup T. Haplogroup K is spread all over Eurasia, Oceania, and among Native Americans. K is found mainly in Melanesia, Australian Aborigines, India, Polynesia, and island Southeast Asia. To put it simply, haplogroup K is found in the Oceania region, which includes Melanesia, Australia, India, Polynesia, and Southeast Asia. Haplogroup M. The geographic distribution of haplogroup M is restricted to near and remote Oceania and eastern Indonesia. The majority of males with Papua New Guinea and Melanesia carry haplogroup M chromosomes. Haplogroup S chromosomes are predominantly found in Oceania and Indonesia. No examples of the basal paragroup K2b1 have been identified. Males carrying subclades of K2b1 are found primarily among Papua peoples, indigenous Australians and Polynesians, etc. Its primary subclades are 2 major haplogroups: haplogroup S, also known as K2b1A, found in highlands of Papua New Guinea, and haplogroup M, also known as K2b1B, found in New Guinea and Melanesia. So to simply put it, haplogroup M and S is found in Oceania, distributed among peoples of Papua New Guinea and Australia. Haplogroup Q. Haplogroup Q is distributed widely in North Eurasia and is found at high frequencies in some Siberian groups. and at low frequencies in Europe, East Asia, and the Middle East. It is also the major lineage among the Native Americans, with Q-M3 being almost completely restricted to the Americas. Undifferentiated Paragroup Q is observed at low frequencies in India and Pakistan. Q1A1 found with low frequency among Han Chinese, Japanese, Koreans, Mongolians, Tibetans, etc. Q1A2 found at low to moderate frequencies among populations of Southwestern Asia, Central Asia, and Siberia. Q1A star found at low frequency in Pakistan, India, and Tibet. Q1A3A typical of indigenous peoples of the Americas. Q1A3A1 found among some indigenous peoples of South America. Q1A6 found in a significant minority of Yemeni Jews. To put it simply, haplogroup Q is observed and found in Asia and the Americas, with Q-M3 being in North America, Central America, and South America. Haplogroup R. The majority of European Y chromosomes belong to this clade. Haplogroup R is defined by the SNP-M207. The bulk of haplogroup R is represented in descendants of subclade R1, which likely originated on the Eurasian steppes. R1 has two descendant subclades, R1A and R1B. R1A is associated with the Proto-Indo-Iranian and Balto-Slavic peoples and is now found primarily in Central Asia, South Asia, and Eastern Europe. Haplogroup R1b is the dominant haplogroup of Western Europe and also found sparsely distributed among various peoples of Asia and Africa. Its subclades R1b1a2 is the haplogroup that is most commonly found among modern Western European populations and has been associated with the Italio-Celtic and Germanic peoples. R1 found throughout Western Eurasia, R1a found in Central Asia, South Asia, and Northern and Central and Eastern Europe and the Balkans. Haplogroup R1b found in Western Europe, West Asia, Central Asia, North Africa, and Northern Cameroon. Haplogroup R2 found in South Asia, Caucasus, Central Asia, and Eastern Europe. To put it simply, haplogroup R is found in Central Asia, South Asia, Central, Northern, and Eastern Europe, Caucasus, Central Asia, South Asia, North Africa, Central Africa, and various other places. So as stated before, haplogroup CF is represented best by Japheth. Haplogroup CF is mostly Eurasian lineages in the world. It would also appear that most of Japheth's descendants are lighter skinned. They also seem to have settled mostly the northern parts of the world. Now let's shift our attention to Shem. I believe Shem is haplogroup DE. Haplogroup DE is a human Y chromosome DNA haplogroup. It is defined by the single nucleotide polymorphism SNP mutations YAP. DE is unique because it is distributed in several geographical distinct clusters. An independent subclade, haplogroup D, is mainly found in East Asia, parts of Central Asia, and the Andaman Islands, but also sporadically in West Africa and West Asia. The other immediate subclades, haplogroup E, is common in Africa and to a lesser extent the Middle East and Europe. According to the paper titled New Binary Polymorphisms Reshape and Increase Resolution of the Human Y Chromosomal Haplogroup Tree, and it reads, haplogroup D chromosomes have not been found anywhere outside of Asia, the likely place of origin of this haplogroup. D lineages are most commonly found in Central Asia, Tibet, and in Japan, and are also present at low frequencies in Southeast Asia and among Andaman Islanders. Haplogroup E. This makes haplogroup E by far the most mutationally diverse of all major Y chromosome clades. These polymorphisms define 56 distinct haplogroups, which can be found at high frequencies in Africa, at moderate frequencies in the Middle East and Southern Europe, and with occasional occurrences in Central and South Asia. Early human populations that can best be represented by the descendants of haplogroup DE would be the basal Eurasians, because haplogroup DE is an early Eurasian lineage. Frequency of Y-DNA haplogroup E in modern populations and hypothetical migration route from its ancestor, clade DE, in Southwest Asia, the Middle East. Haplogroup E arrived in Africa about 77,000 years ago and diversified in the Horn of Africa. It arrived together with specific West Eurasian alleles and later also diffused into various Paleo-African populations, as well as among the ancient North Africans, which were a population indigenous to Northern Africa and distinctly related or ancestral to modern West Eurasians. Haplogroup E and DE have a Middle Eastern origin. Haplogroup E migrated into Africa as part of a mass migration of basal Eurasians, ancestral West Eurasians, and then diffused and diversified within Africa, specifically the Horn of Africa, Northeast Africa. Haplogroup DE, next to CF, were the basal Eurasian lineages which expanded from the Middle East. The common ancestral lineage Y-DNA atom is suggested to have originated somewhere in the Middle East or the Iranian plateau. Massive Eurasian migration and following admixture created the many different African populations of today and are the reason for the high genetic diversity observed in sub-Saharan Africa. In other words, modern African people, sub-Saharan Africans, form from indigenous Paleo-African groups and intrusive Afro-Sans, basal Eurasians and ancient North Africans. Finally, there is the fact why haplogroup E is dominant among non-hunter-gatherers in Africa and is within the Eurasian subclade DE, while indigenous African hunter-gatherers belong to haplogroups A, A00, and B. Haplogroup E actually back-migrated to Africa and overpowered the indigenous populations of Africa, which is haplogroups A and B, which would be the Hamites. This is why haplogroup E is considered to be Afro-Mediterranean. A new study concluded that there was a massive migration of non-African Eurasians back into Africa about 70,000 years ago, spreading the Eurasian haplogroups such as E and its parent DE within Africa, before additional waves of Eurasian admixture. Modern Africans are thus descendant of Paleo-Africans, which is haplogroups A and B, and Afro-Sans, which is haplogroup E, which merged in Africa between 70,000 years ago and 40,000 years ago. According to the paper, carriers of mitochondrial DNA microhaplogroup L3 basal lineages migrated back to Africa from Asia around 70,000 years ago. It reads, the split of the Y chromosome composite DE haplogroup is very similar to the age of mtDNA L3, and Eurasian origin and back migration to Africa has been proposed for the African Y chromosome haplogroup E. This correlation rather seems to be the result of a joint and global replacement of the old autochthonous male and female African lineages by the new Eurasian incomers. Basically, haplogroup E was a Eurasian lineage that entered into Africa, replacing the autochthonous or the indigenous African populations. because haplogroup A and B is considered to be true African or Paleo-African, whereas haplogroup E is considered to be Afro-Mediterranean. It came into Africa and began to conquer the region. So Shem's descendants are believed to have settled both Asia and Africa. Shem would have been the father of Afro-Asiatic Semitic populations. According to Britannica, a Semite, a member of a people speaking any of the group of related languages presumably derived from a common language, Semitic. And according to Britannica, Semitic languages that form a branch of the Afro-Asiatic language phylum. Members of the Semitic group are spread throughout North Africa and Southwest Asia and have played a prominent role in the linguistic and cultural landscape of the Middle East for more than 4,000 years. And according to thefreedictionary.com, Afrosan, a large member of the related languages spoken in Asia and Africa, Semitic, a major branch of the Afro-Asiatic language family. And as we can see, Afro-Asiatic languages are spread in both Africa and Asia, but it's the Semitic family of Afro-Asiatic that is seen in both Asia and Africa, the most widespread of the Afro-Asiatic languages. Therefore, I believe Shem is haplogroup DE, since Afro-Asiatic populations descend from this haplogroup, more specifically haplogroup E-P2 or EV38/M35. According to the book In Hot Pursuit of Language and Prehistory, it reads, most of the Afro-Asiatic speakers shared the lineage defined by Yap descendant called PN2-215-M35. And according to the book titled The Genetics of African Populations in Health and Disease, it reads, for example, Underhill et al. 2000 state that the V38/M35 lineage was carried into the Near East from Africa in the Mesolithic. This V38/M35 in Africa corresponds to the TAQI49AFV. It is far more likely that pre-Proto-Semitic speakers went into the Near East at this time, where Semitic languages have long since dominated. And according to the paper titled Y Chromosome E Haplogroups, Their Distribution and Implication to the Origin of Afro-Asiatic Languages and Pastoralism, it reads, the Proto-Afro-Asiatic group carrying the E-P2 mutation may have arrived at this point in time and subsequently gave rise to the different major population groups, including current speakers of the Afro-Asiatic languages and pastoralist populations. Haplogroup E actually overlaps quite nicely with Afro-Asiatic populations, as we can see. But haplogroup DE is a unique lineage from all other Y DNA chromosomes. Haplogroup DE has a specific gene that haplogroup A and B and CF doesn't have, called YAP. Only people who descend from haplogroup DE has this unique genetic marker. The most well-known unique event polymorphism that defines DE is the Y chromosome allele polymorphism YAP. The mutation was caused when a strand of DNA called an allele inserted a copy of itself into the Y chromosome. Hence, all chromosomes belonging to DE, D, E, and their subclades are YAP positive. All Y chromosomes that belong to other haplogroups and subclades are YAP negative. According to the paper titled YAP Signature of an African Middle Eastern Migration into Northern India, it reads, YAP, an allele insertion polymorphism found on human Y chromosome, is present in two lineages worldwide corresponding to haplogroup E and haplogroup D polymorphisms respectively. First lineage belonging to haplogroup D is specific to Japan and other Southeast Asian populations, while haplogroup E is confined to Sub-Saharan African, Middle Eastern, and Southern European populations. This YAP-positive marker that only people who descend from haplogroup DE have, to me gives more credence to 3 ancestral nodes for Shem, Ham, and Japheth, since haplogroups A and B and CF are YAP negative. It makes sense to divide the 3 haplogroup branches as such. Haplogroup DE is perfectly in the middle of haplogroup A and B and CF lineages. With that being said, the model I assign is this. The African haplogroup A and B is Ham, the Eurasian haplogroup CF is Japheth, and the Asian and African haplogroup DE is Shem. I end with this. We all descend from Noah, a righteous man, a great prophet, and a man chosen by the Most High.
But Noah found grace in the eyes of the Lord. These are the generations of Noah. Noah was a just man and perfect in his generations, and Noah walked with God. And Noah begat 3 sons, Shem, Ham, and Japheth.
And with that being said, be blessed and shalom.